Good afternoon, everyone, and welcome to the MassCONNECT Holiday Showcase. I'm Jason Quadiro, Vice President of Business Operations here at MassBio. So excited to welcome everyone today, including our presenters, our sponsors, Boehringer Ingelheim and Evaluate, as well as our mentors who have dedicated so much over the course of this year. Today is our final MassCONNECT event of the year. And what a year 2020 has been. So challenging for everyone, including ourselves here at MassBio. But I think for us and for the industry, it's also been quite rewarding. It's been really rewarding to be a part of this industry who's been so key in solving this global pandemic, whether through diagnostic development, therapeutics, and now the vaccine. It's been just quite the year for the life sciences as a whole. So really rewarding for MassBio to continue to help foster early stage company development and innovation in Massachusetts. We're also happy with the amount of new members that we've had this year. MassBio added over 180 new members. We've saved millions of dollars through the MassBio Edge programs. And we're also launching a new program called Partnering Week that you should all be, interested in. At the end of January on the 25th, we have 4 major sponsors, and we're going to continue to help make introductions for these emerging technologies and companies to the large pharma. So really excited about Partnering Week and what we're doing with the Innovation Services team moving forward. We're also able to pivot MassCONNECT to a fully virtual platform. And in doing so, we still had the most program graduates ever. 18 in total, with participants from all over the country and the globe, including Israel and Belgium. I think that's just a real testament to the hard work of Rachel and the dedication that she's put in, the time and effort. So, really applaud her and thank her and the team for all the hard work. We had a record number of mentors through the course of the year, over 125 volunteers with over 1,500 hours of your time. So thank you for that dedication. We launched our first successful spin-out of MassCONNECT, MassCONNECT.dh. This was launched this year with the sole focus on digital health innovators. And we had 8 graduates from that program in its first year. Just again, incredible work from Rachel and the team. None of this would be possible without the incredible support from all of our mentors, the project managers, and of course, our sponsors. So thank you again for all the dedication and hard work. I want to congratulate our 5 graduating companies today: PrimeVax, Mark Therapeutics, LIMAX Bio, Watershed Informatics, and Step Up IC. We're so thankful to have been a part of your journey, and we really look forward to your continued and future success. We're going to be along there for the ride. And we hope to see everybody in 2021. Hopefully things go back to normal. It is the holiday season. It is a time for hope. And we are hoping that with all the hard work this industry has done, things are going to start to turn around sooner rather than later. So while we're virtual today, by the end of 2021, we hope to be getting back together in person. Wouldn't that be incredible? But in the meantime, we look forward to seeing everybody at tomorrow's virtual holiday party for MassBio and MassCONNECT. If you want, head over to massbio.org to register, and we would love to see you guys all tomorrow. So again, thank you to everyone, and to the graduates, congratulations. And I'll kick it back over to Rachel.
Excellent. Thank you, Jason, and welcome everyone. Let's see if I can there we go. So today we'll be listening to the 5 companies that are graduating from the final cycle of MassCONNECT this year. Typically, after these presentations, our super mentor Pravin would be opening the bar downtown at Cooley's office in the Back Bay. This year is obviously a little bit different, but we do hope that it's just as festive. So we have a lot of content today. So we're splitting up today's event into 2 parts. We'll have the holiday networking tomorrow, like Jason just said, at 4 o'clock in conjunction with our MassBio Holiday Zoom Mixer. If you haven't had the chance to register yet, please just ping me and I can register you on the back end. But you should know that if you're going to come to the holiday mixer tomorrow, we are expecting you to wear an ugly sweater or some reindeer antlers. So before we jump into the presentations, I do want to give those that are joining us for the first time just a little bit of context. The 5 life sciences startups that you'll be hearing from shortly were hand-selected to participate in the MassCONNECT and MassCONNECT DH program at the beginning of the fall. In October, they presented their technologies and business plans here over Zoom. They told us what they needed help with to bring their ideas forward. After those initial presentations, we had 58 mentors step forward to volunteer their time and help these companies refine their strategies, pressure test their ideas, and have a larger understanding of how the biopharma world evaluates these incoming technologies for potential collaboration or investment. Each entrepreneur here worked with a team of 10 mentors over the course of 6 to 8 weeks in weekly 1-hour group sessions and countless hours offline, one-on-one. Today is a culmination of everyone's efforts, and you will be seeing the updated strategies and business plans for these 5 companies. We would not have been able to do any of this without the incredible support of our community. I do want to thank our first wonderful sponsor, Boehringer Ingelheim, and also Evaluate, that have been extremely instrumental with their support and their resources for our companies and our program. It was an absolute pleasure to work with the BI team, Scott, Monica, Marcio, and Mark. I was so impressed. Everyone on the team was so dedicated to our entrepreneurs, and you were all such an asset during our mentor meetings. I'm now pleased to welcome Scott Dwyer, Executive Director, U.S. Head of Business Development and Licensing, to say a few words on behalf of Boehringer.
Great. Thank you, Rachel, and thank you, Jason. You can see I'm already in the festive mood for tomorrow's party, and I'm going to have to dig out a different sweater for tomorrow now, I guess. And I'm also located a few hours east of you, and you can see the snow has already hit that's headed for the Cambridge area tonight. But yeah, this program has been great. So on behalf of Boehringer, this has been such a positive experience. You know, we're proud to sponsor biotech community and proud to sponsor this innovation in the Cambridge area and all throughout the world. This was just another opportunity for us to learn something ourselves and to help these small companies. Faye, Monica, Mark, Marcio, and I all participated as mentors, and I talked to each of them today. And we all took something great away from this program. The panel of mentors that you were able to put together was quite impressive for each company. So congratulations on having such a great network and being able to tap into all that local talent. We found great networking opportunities for ourselves. I know, speaking to Mark from our venture fund, he's already had some follow-ups with some of the other mentors and made some really good connections. Positive experiences all around. To watch these CEOs who have so much talent grow and respond to the feedback and to see the pitches improve over time. If I have to summarize the experience, at least on the therapeutics track, in just a couple of words, I would say very daring. You know, you have these 3 companies that are all trying something kind of amazing. One of them taking a deadly human pathogen and trying to redirect it towards cancer. Another one taking a virus and putting it in the brain of Parkinson's patients. And then another one trying to disrupt the wound care and surgical adhesives field with natural products. It's really, really cool. And I know there's the same level of innovation on the digital side as well. So thank you to MassBio and MassCONNECT for having this program. We're happy to sponsor it. And it was just such a pleasure to take part. I'm looking forward to some great pitch presentations. And I hope you all have a great holiday, prosperous new year, and enjoy the presentations. Thank you.
Thank you, Scott. And I'm so sorry, I forgot Faye. Faye, you were also instrumental, and she even helped me with the selection of these companies here today. So thank you as well, Faye, and thank you, Scott. I also want to thank Sean Crowley, Director of New Business Development at Evaluate, for your support of our program this entire year and giving all of our participants licenses to the Evaluate platform during the program. This was so critical for our entrepreneurs who were diving into their market research and refining their value propositions amongst the competition. You might even see some Evaluate references in the footnotes of some of the presentations today. Sean, over to you for a few words from Evaluate.
Thanks, Rachel. So good afternoon, everyone. As Rachel said, my name is Sean Crowley. I'm here representing one of the program sponsors, Evaluate. So first off, would like to thank MassBio, Jason, Rachel, everyone on the Innovation Services team for putting together another phenomenal cycle of MassBio with an exciting and diverse set of companies to round out 2020. Really looking forward to the final presentations today and seeing all the progress that's been made over the course of the last 8 weeks. So at Evaluate, we're really proud to continue the relationship with MassBio and our sponsorship of the MassCONNECT program. We feel that the program is integral to the health of the ecosystem in Massachusetts and really provides a unique opportunity for early stage companies to work directly with industry mentors and gain valuable insights to help them advance and reach their goals. So very much look forward to continuing to follow the progress of the participating companies who are now joining the ranks of the many successful MassCONNECT alumni as they look to pursue their full potential, deliver innovative therapeutics, diagnostics, and devices to the patients that need them. So during the course of the program, the participants had access to our Evaluate Pharma and Evaluate MedTech market intelligence platforms. I'm hoping to see, Rachel, a lot of those that data cited in footnotes. And for those of you who aren't familiar with our platform, they would have used our content and our data to support a number of different exercises. So from sizing their markets of interest to monitoring the competitive landscape to identifying potential partners, analyzing comparable deals and transactions. With that said, very much looking forward to the program today. The MassBio holiday party tomorrow. And Rachel, I'll pass it back to you, but thank you again for the time and for running this wonderful program.
Thank you, Sean. And thank you both. I also do want to thank Chandra Ramanathan and our friends at Bayer for their support and their sponsorship for this event today. So next, let's see if I can also, here we go. I want to thank our incredible mentors who joined us this year, gave back their time, their early mornings, their late nights, the one-on-one calls, the insight, the advice, the tough love, the introductions, and the ongoing support. It has been such a pleasure working with you all, and I think I have learned as much as our entrepreneurs have from you. And I am especially grateful to you all for your willingness to have an impact on the trajectory of these entrepreneurs' journeys. And I hope you have all found the experience rewarding. Lastly, I want to thank our wonderful project managers, Rachel, Roma. Thank you for coming back and being our seasoned rock stars. And it was also great to work with Yael, Mark, and Shu Wen and get to know you all. You kept us all on track during our meetings and were a huge, huge part of this program being a success. So without further ado, I am pleased to start the presentation component of the hour. We will have 5 presentations total. Again, each entrepreneur has 10 minutes to present. Afterwards, I will be relaying any questions from the Q&A function. If you hover to the bottom of your screen where it says Q&A, that button, you can put your questions for the team there. We will have roughly 5 minutes for questions after each presentation. And without further ado, our first presenter is Tony Chen from PrimeVax Immuno-Oncology. Over to you, Tony.
Thank you so much, Rachel. And thank you very much again to the MassCONNECT team and everybody involved in helping us get this going so far. Let me share my slides and we'll get right into it. Can you guys see this now? All right, very good. All right, great. Thank you guys so much again. Excited to share our progress. So PrimeVax is a clinical stage cancer biotech using a novel dengue virus to treat cancer patients. And so first, to set the stage, Dr. Kaufman, who was instrumental in getting the first FDA-approved oncolytic virus, summarized it really well. Oncolytic viruses trick the immune system to think that they are actually tumors, that the tumors are actually virally infected cells. And while they've been studied for over 50 years, really only in the last 5 years, oncolytic viruses have actually emerged as one of the most exciting classes of immuno-oncology. From a development perspective, there are now over 100 OV clinical trials, both in mono and combo therapy. And from an investment perspective, I think we see a lot of excitement across the board from pharma and investors with significant deals announced in just the last 5 years. And circling back to Dr. who has been at the forefront of much of this OV development, we are very humbled to be working with him in our clinical trial. So that brings us to PrimeVax. We are a clinical stage immuno-oncology biotech, and our lead asset is the dengue virus, which we are targeting late-stage cancer patients. So our early data suggests highly novel mechanisms of action, which I will cover shortly. We have an IND opened, and the FDA has already blessed our clinical strategy. Our phase 1A human safety data of 12 healthy volunteers showed a well-tolerated profile, which is further supported by the Army's 20 years of historical data with our strain as well. We have a very unique competitive positioning. Our license with the US Army allows us to use their dengue virus vaccine strain to treat all cancers worldwide. We wholly own 9 patent families in dengue. And we've surrounded ourselves with a rock star team of experts and development partners. We've raised $4 million to get to where we are now, and we are now raising a Series A to advance clinical studies. So our lead asset is dengue virus serotype 145AZ5. It is an attenuated strain of dengue created to induce a safe yet tolerable fever. The virus has 3 main mechanisms of action: intratumoral, multiple immune effects, and a drug-like effect. In the coming 3 slides, I will dive into each one. So first, it's an oncolytic viral effect. The dengue virus directly infects and kills tumor cells intratumorally. We know that it's the HSPG receptor, and literature on the right shows 5 different tumor cell lines that all share the same vulnerability to HSPG infection. And in literature, we see another 10 plus different tumor types that are also sharing the same vulnerability. Using heparin, which is a protein compound, to cleave the HSPG receptors, what we see is that our strain of dengue, 45AZ5, drops its infectivity ability by 50%, even with a tiny amount of heparin. So that shows us that our dengue virus is highly dependent upon HSPG to infect tumor cells. Moving on to effect number 2, when we actually think of dengue, we think of dengue causing a powerful fever, right? Dengue fever. And the data on the right mimics what the human biological system actually undergoes in reaction to the fever. So first, our phase 1A data showed that an infection of the blood caused high activation of the transcriptomes and a broad range of cell populations, such as T cells, NK cells, myeloid cells, etc. Next, downstream immunologically, what we see is that the proteins responsible for these interactions have actually shown an increase in the expression of class 1 MHC on tumors as well. And those same tumors, when you apply them into a tumor killing in vitro assay, we showed superior tumor killing because of the influence of the dengue virus specifically on the tumors. We see both T cells and NK cells both being killed faster because of the dengue. And effect number 3, this is what our advisors really call our wow. So here, the supernatant secreted by the blood during an active dengue infection actually shows killing of tumor cells without the need for T cells or NK cells. So now it appears that the virus actually behaves like a drug as well. And our data on the right shows that 60% of the tumor killing in a refractory melanoma cell line occurred in 6 hours. And we showed similar tumor killing in breast, lung, and another melanoma cell line as well. And the early understanding of why we think this is happening is that it's most likely a trail and TNF-induced pathway mediated by interferon alpha response. We observed dengue fever activates these. We will confirm these further in our upcoming preclinical studies to really drive it, more of the why behind the mechanisms. So bringing it all together, we see that dengue virus attacks cancer like a virus. It has a multiple immune stimulatory responses, and it also has like a drug-like tumor killing effect. And so we're not aware of another virus that can actually do all 3, and certainly not with the same known safety profile that ours has. And so speaking of safety, we actually have phase 1 data on 12 healthy patients, and we have 26 patients of data from the US Army. And the short soundbite at the bottom here really summarizes it. Dr. ND concludes that the dengue virus 1, our 45AZ5 strain, was well tolerated and resulted in an uncomplicated dengue illness and may be a suitable model, for therapeutic product testing. The patient journey from an infection perspective is about 28 days. You get injected on day 0, incubation for 9 days. The fever phase is day 9 through day 15, and then the patient recovers back to baseline by day 28. The adverse events are here, SAEs as a percent of AEs is around 7%. And most importantly, all adverse events resolve by day 28. And so we and the FDA at this point conclude that we feel very comfortable with the safety profile of our dengue virus. And turning to where we are, in the last 5 years, we have taken the lead asset, PB001DV, from discovery to IND and then to phase 1A. Our IND will initiate monotherapy in late-stage melanoma. And in addition, we're going to do preclinical data in other solid tumors as well. And as we progress, we're going to evaluate our lead asset, dengue, as a powerful combination therapy, not only with checkpoint inhibitors, but also with next generation dendritic cell therapy as well, which is another product in our pipeline. And turning to our development plan for the next 2 years, we're laser focused, of course, on hitting our milestones, which is really to get our PB001DV into treating cancer patients. That's the next stage. We're currently raising $15 million to be able to support a series of advanced mechanism of action preclinical models, as well as to initiate our melanoma patient trials. And with the funds, we'll be able to treat 15 patients in refractory melanoma, as well as support product development for additional indications and combo therapies as well. This plan positions us really well for exciting partnering and further development opportunities in the industry. Inspirationally, we've continued to build a network of amazing partners in drug development. PrimeVax was founded on the West Coast, and like most early stage companies, we've operated virtually for the last year. But as you can kind of tell, our partners are really on east of the Mississippi. And over the next 6 months, we're probably going to relocate the company a little bit further east to optimize, like the operations and to meet our corporate objectives. I do want to cover the patents and the license briefly. Our patents on the left here cover the use of dengue virus in all cancers worldwide. Our patent strategy was to corner the dengue plus cancer market as a monotherapy as well as a combo therapy. We've secured 9 patent families worldwide. And our license from the Army on the right for 45AZ5 offers us a unique competitive position, which not only gets us the worldwide exclusive license from the Army for development and commercialization for all cancers using their dengue, but also the product rights and the data rights to purchase the cGMP product, and the fact that the CMC data is already on file with the FDA for a clinical grade ready to inject product into cancer patients next. Turning over to team, I want to speak to our team here. We've been operating in a very lean business model so far. Myself and Bruce as co-founders, we've been at this for about 5 years, building it from inception to IND. I have a mixed pharma consulting and commercialization and healthcare background. I worked at a CRO. I wrote the value messages for Opdivo, among other things, as well as implementing the Affordable Care Act for the Department of Health and Human Services. Bruce, my co-founder, led the early R&D, and without him, there would be no PrimeVax. He's the inventor and really the original spark of brilliance that kind of came up with the idea of using dengue as an oncolytic virus. And Gita, who's been on the call here with me today, has joined us recently. Gita is a seasoned executive with over 25 years of cross-functional experience in corporate development and strategy, and really has been helping drive PrimeVax as a company through like an external lens. And we've recently, really just in the last 2 months, brought Dr. Andy Zloza on board as a part-time basis to head up translational science for us. And with funding, we will look to expand the clinical operations team with a chief medical officer and a VP or director of clinical ops. Our board of directors include Dr. Bernie Fox and Dr. Franco Marancola, who are both ex-SITC presidents and respected immuno-oncologists. Speaking to the select advisory team, Primevax is very honored to be working with the foremost experts in dengue virus, like Dr. Stephen Thomas on the left, and Dr. Nina Bardwaj, who's the foremost expert on dendritic cells. Most notably, we have the 4 out of the last 5 presidents of the Society for Immunotherapy of Cancer, either as board members or collaboration partners working with us. And rounding it out, we have other SAB members with extensive track records in developing multiple immuno-oncology therapies as well. Wrapping up, I think we've taken a very prudent approach to getting this where we are. We've de-risked the science and de-risked the investment. We really focused on hitting our milestones. Our IND is opened. We have a strong IP portfolio. We've generated compelling data, both safety, human, and some early preclinicals. And we've been operating very leanly. We're now raising $15 million to advance dengue into the clinic for the first time worldwide. Thank you very much for listening. And most importantly, thank you really again for the opportunity to be here with the MassCONNECT cohort and to our illustrious mentor team that you see here. Thank you so much again.
Thank you, Tony. Excellent. So we will now open it up for questions for Tony, and also his CBO Geeta is also joining us. Please, I would like to direct you to the Q&A function that's at the bottom of the Zoom window. And you can type your questions there, and I will be selecting and relaying back to the team. No open questions yet. Blew them all away. I'll also read comments as well. Comment from MassCONNECT mentor Leora Schiff. Tony, really nice presentation. Congratulations.
Thank you very much, Leora.
All right, first question is coming from Anonymous. Nice presentation. Any potential issues in subjects with previous exposure to dengue virus?
It's a very good question. What we are actually doing in our upcoming clinical trials for cancer patients is to screen them out for dengue naive patients first. And that's the vast majority of patients in the developed healthcare markets, such as the US, Canada, and Europe. Yes, definitely it is a potential concern. We do not want patients who have already been exposed to dengue in the past. We want that naivete to help convert some of that immune memory, because dengue does have a very, very powerful immune response and memory response.
Wow, thank you. Next question. Moving forward, what type of partnerships and associations are you looking for?
Yeah, so I think that it's a great question. It's something that I think we'll always evaluate on an ongoing case-by-case basis. But the high level is that we are looking to take. We've been very good at focusing on internal, hitting our milestones for the last 5 years. But now I think what we want to be doing is collaborating with their biotechs, with pharma companies, with like nonprofits, other academic shops, as we build out the clinical program as a next step.
We'll leave it open for just a few more seconds.
Sounds good.
I think, Tony, you were just very succinct in your presentation.
You know what? That's what we aim for, right? Sometimes less questions is better.
Just a few more seconds so we can read them live. I can see someone's typing. Okay, question from Anonymous. What makes dengue virus unique?
Dengue virus is unique in the sense that it causes a very, very strong whole body fever. In the context of oncolytic viruses, most other oncolytic viruses are not built with the intent of actually causing a febrile effect that is acute and episodic in nature. What we've observed is that there's powerful memory responses coming out of the dengue after the dengue infection is over. And what we want to do is redirect that toward the cancer. Anecdotally, what we've also seen is that the transcriptomic analysis of, like actual increases to cell populations during a dengue infection, at a very cursory high level are, something to the effect of, 10 to 100x higher than the other oncolytic viruses in the space. Dengue also causes a much broader immune response than most other oncolytic viruses based on cursory initial literature search.
Excellent. All right, Tony, that is it from the audience. Congratulations. Wonderful job. And then if anyone from our audience thinks of additional questions later, they might pop into your head, feel free to use the Q&A function and Geeta or Tony can answer there. Thank you.
Absolutely. Thank you again.
Wonderful. Our next presenter is Victoria Bolotina from Mark Therapeutics. Over to you, Victoria.
Unmute myself. So now I'm unmuted and I'm sharing the screen, correct?
Yes, if you just want to go into presentation mode.
Yep.
Perfect.
And I would like to start my presentation with a great thank you to MassCONNECT and for the opportunity to join this exceptional training program. And special thanks to Rachel, the director of this program, and to Rachel Cotton, who was the project manager for us all this time. And I would like to thank very much all our mentors, all 13 mentors together, and each of them individually for their enthusiastic support, expert advice, and for all the zillion of great questions and suggestions which they gave us during these 8 weeks of general meetings as well as offline personal meetings. Thank you again, all guys. You are great, and I'm very happy to have your continuous support of this program. I'm happy to present Mark Therapeutics, which is championed by the spectacular group of business and Parkinson disease experts. First of all, my co-founder and champion of the program, John Ripple, who is the chair of our board of directors. Thank you also for John Tiglimonte, whom you know, most of you, who is advising our company. And also I would like to introduce Jeff Cordover, who is one of the world leading experts in Parkinson disease and who has already dozen human and preclinical as well as human trials in Parkinson field. All together with this group, we are developing gene therapy to fight progression of specifically idiopathic Parkinson disease. And Mark's fight against Parkinson disease is personal for me. The company bears the name of my father, Mark, who was one of too many. It's named after my father, who's one of those Parkinson disease patients who was devastated with this disease. And most of you must probably know someone with this disease and know that it is caused by premature loss of very specific dopaminergic neurons deep in the brain. And what most probably many of you don't know, that more than 80% of all Parkinson cases are idiopathic, also called sporadic PD. Which don't carry any genetic mutations. Those patients don't have any family history, And currently there is absolutely no way to predict who will get this disease when they will be aging. And that's one of the problems of this disease, why it is remains so problematic for treatment. First of all, the main problem is that after the person is diagnosed, clinically diagnosed with Parkinson disease, which is usually happens when they start to develop slight tremors or some other motor dysfunction, there is the inevitability of progressive death of their dopaminergic neurons going forward, which leads to progressive progression of debilitating motor dysfunction, as you can see here in red, growing sharply upwards, showing this increasing number and severity of the symptoms, which is soon followed by a lot of complications, behavioral complications, and ending with premature death. And the main problem is that for all these patients is that there is no therapy which can currently halt or even slow down the progression of this disease. We believe that we found a game-changing solution which can halt this progression. And this solution is based on long sequence of experiments and studies, mechanistic studies, which lead to the finding that all idiopathic PD patients, which we tested so far in 3 small groups of patients, lose expression of one key enzyme. And this enzyme is critical for cellular function, which is calcium signaling in the cells. And loss of this function can trigger major pathological processes involved in PD. And we also found that progression of this kind of deficit and Parkinson, leading to Parkinson disease, can be still halted by one-time AAV gene therapy that we have already plenty of evidence can restore protein expression, fix its function, and stop progressive loss of the dopaminergic neurons in the brain, and effectively stop progression of the disease, which we hope will be happening in the patients in the same or similar way as we already see in preclinical trials and mouse models. So we have plenty of data, and I don't want to abuse you with going through all mechanistic studies. All what I want to tell you today is that our treatment, this Mark 101 treatment, in fact, stopped progression of the loss of the dopaminergic neurons when we tested it in a new idPD-like aging mice model. So on the right, you can see those dopaminergic neurons in this mice, and in the normal control mice representing healthy humans, and in PD mice, which represents the idiopathic PD humans, and the treatment with Mark 101 fully stop progressive loss of those dopaminergic neurons in this model. As a consequence of this halting progressive loss of dopaminergic neurons, we also saw halted progression of PD-like motor dysfunction in these animals. And on the right, you can see example of the motor dysfunction developing in this idiopathic PD mice. untreated or treated with Mark 101. You can see that it is in fact stopping progression of this dysfunction, and we found, we tested it in aging mice, and we found that this treatment is safe. So what is so special about Mark Therapeutics? Why you should look into this very carefully and support its further development? So current standard of care right now focuses on symptomatic treatments only, meaning that there are multiple treatments and they are developed and still a lot of companies are finding better and better treatments that can help with symptoms for those patients. But the holy grail of the treatment, which is not there yet, is disease-modifying treatments, which can stop progression or somehow slow progression or can in any way modify the cause of this disease. And most of the companies do it using small molecules or antibodies, which are right now in different stages of development. So the newly emerging therapy and very exciting new direction for Parkinson patients is gene therapy, which is one-time and target-specific therapy, which all the patients are really are waiting for. And you most probably, most of you heard, especially in view of most recent news about PREVAIL, success of PREVAIL, which is using gene therapy as one-time target-specific treatment for familial PD patients with GBA mutations, which is less than 10% of all PD patients. And here is Mark Therapeutics. We're using a very similar approach, gene therapy, one-time target-specific approach, but we're targeting, as I already mentioned, more than 80% of PD cases because we're targeting idiopathic PD patients. And the difference, major difference between all existing methodologies and treatment options right now are that all of them are mostly targeting classical PD molecules, while Mark Therapeutics found and is targeting novel molecule, that's PLA2 group 6L enzyme, which is encoded by Park14 gene. And its epigenetic deficit is found in idiopathic PD patients. And one of its great advantages that this target is located upstream from all other classical targets and can cover idiopathic PD patients and stop progression of this disease there. So we already completed a lot of studies related to target validation, biomarkers, efficacy and safety demonstration in mouse and human cells using support of Michael J. Fox Foundation and others. And now we are looking for seed money, seed financing for preclinical optimization and which will include in vitro and in vivo studies. And I will bring my presentation to conclusion with simply leaving you with this executive summary, which highlights the new target of Mark Therapeutics. And we have all information about the mechanism of this target involvement in the idiopathic PD pathology. We developed this one-time gene therapy to correct this deficiency and restore important cellular functions. And we, based on our preclinical animal model studies, we expect therapeutics outcome to result in halting progression of this disease. The IP is covered by issued patents, and we are seeking capital for preclinical IND enabling studies. Thank you very much.
Excellent. Thank you, Victoria. We will now open it up for questions in the Q&A box. I disabled the chat just so I could only read one thing. First question for Victoria is from our MassConnect mentor, Eric Evans. Excellent presentation, Victoria. Can you say more about the use of the $5 million seed proceeds?
I will, as I already mentioned, we can go here. This 5 million will be spent for preclinical optimization for in vitro biomarkers, AAV development, and in vivo optimization, delivery, dosing, efficacy in at least 2 mouse models and preliminary toxicology.
Thank you. And comment from your MassConnect mentor, Arthur Hiller. Well done, Victoria.
Thank you.
Let's see. Question from Anonymous. Are you looking for any regulatory partners?
Yes, we're looking for regulatory partners who can help us bridge the preclinical models and the readouts in those models with eventual anticipated clinical trials and the readouts, which will be quantitative readouts, which we'll be using in actual patients. And we will appreciate help in identification of all the particular experiments and tests and data points which we'll need to have in hand for IND application.
Great. All right, just a few more seconds open. Oh, we got quite a few. Question from Christine S. Can you talk about the method of drug delivery?
We are trying several routes of delivery. It will be direct delivery into the brain, but via different routes at the moment, and we will be picking up the best, the most effective and specific route. And as you know, the direct delivery into the brain is not, rather than, well, has multiple advantages in comparison with more systemic through the systemic administration of this AAV and more targeted and more less material, less immune response, and so that's the way to go. That's the way as the field is now developing.
Thank you. One more question. Have you identified any potential safety issues resulting from modulating PLA in the brain, the specific PLA2G6L?
No, we didn't. And this treatment expression of this particular target in the brain is safe and not expected mechanistically again to cause any problems, because again all we were doing is compensating something that is missing, and that's our preliminary, not preliminary, our preclinical studies show that the mice lives happily until the end of its natural life without any complications.
Thank you. All right, Victoria, you have quite a few more questions in the Q&A, so I will direct you there to continue to answer and to engage with the mentors. But thank you very much for your presentation.
Thank you for your questions.
Excellent. Our next presenter of the afternoon is Ben Friedman from LIMAX Biosciences. Over to you, Ben.
Great. Well, good afternoon, everyone. I'd first like to thank Rachel and MassBio again for the opportunity to participate in this exciting program. My name is Ben Friedman. I'm a co-founder and the CEO of LIMAX Biosciences. Every wound has a face behind its story. Whether it's Adam's scrape or Sarah's chronic pressure ulcer, these are all personal and devastating injuries affecting millions of people around the world. One of those patients is Nick M., whose long-term disability has left him immobile in bed and subject to chronic pressure ulcers. I learned about Nick from his mom, a registered wound care nurse by training, who has spent her entire life devoted to the care of her patients. Sandy would do anything to give her son a better future. But just last year, his sacral ulcer progressed to stage 4, and she has been at his side ever since. Nick is in a pretty serious condition. His dressings don't seem to help, and his last hope at restoring his quality of life involves attaching a cumbersome, bulky mechanical suction device to his wound that requires the assistance of nurses and constant monitoring. Their family wishes that this could have been prevented, but unfortunately, his story is not unique. There's over 7 million Americans living with chronic wounds, which results in nearly $50 billion in cost to the US healthcare system and $25 billion in annual spending. Among these, nearly 3 million patients suffer from pressure ulcers. Together, this results in over 60,000 preventable deaths, $11,000 in average cost spent per patient, and costs exceeding $129,000 for every late-stage hospital-acquired pressure ulcer patient. At LIMAX, we're developing DERMIMAX, a multifaceted solution to prevent early-stage skin ulcers from advancing to stage 4. We've taken inspiration from nature, specifically slug slime, to design patented hydrogel-based materials with strong adhesion to difficult-to-attach surfaces like wet and bloody skin tissues. Our peer-reviewed papers in top journals have shown that this approach can more than double the rate of wound closure in animal studies. DERMIMAX is unlike any other wound care product on the market today. Its tough, stimulating, and dual-layer structure is flexible and is strong to adapt to body movements. It's hydrogel-based to maintain hydration. It contracts at body temperature, it's antimicrobial, and is quick to apply. Our secret is that we use a multi-component proprietary adhesion strategy that dissipates energy at the adhesive interface and maintains strong attachment to tissues by electrostatic interactions, covalent bonding, and interpenetration. Our hydrogel adhesive dressing performs in even the most extreme conditions. Take a bloody and beating pig heart, for example, where it adheres and conforms to the movement of this dynamic and wet organ. But compared to other materials, there's really nothing else like it, with hundreds of times higher matrix mechanical toughness beyond native tissue and adhesive performance well beyond your average Band-Aid and superglue. It's with these unique properties that we believe we can prevent progression to advanced ulcers and reduce patient hospital stays. Through conducting over 120 customer discovery interviews, the National Science Foundation ICOR program this past fall, we've learned firsthand that the ulcer markets are ripe for DERMIMAX innovation. Our target market exceeds $200 million and represents the ulcer, moist, anti-infective, and management market within the US. This is a growing market with many drivers, including increased aging and immobile populations, with existing large companies in this space that provide potential avenues for exits later. Current treatments for pressure ulcers involve frequent wound dressing replacement, tissue debridement and cleaning, and pressure relief spanning multiple classes and types of products. DERMIMAX offers the complete solution to wound management and also takes a preventable approach to progressing to stage 3 and 4 ulcers through its capacity for pressure relief, stretchability and conformability to ulcers, and ability to actively contract and pull wound edges together, similar to a wound vac without the machinery. Over the next few years, we've developed a program development type timeline ranging from R&D and manufacturing, regulatory approval through 510(k) clearance, post-market clinical trials, and commercial development, all requiring additional funding. To date, LIMAX has been supported fully by non-dilutive funding mechanisms through the NIH and Wyss Institute, and we're currently seeking $2 million in seed funding to achieve our next milestones through 510(k) clearance. Through the ICOR program, we've validated that our main customers are surgical centers, wound care centers, and hospitals. And given the large industry and technology adopting market, we plan to sell DERMIMAX as a preventative solution to end-stage ulcers. Importantly, we can produce our materials at low unit costs that may enable competitive pricing as a premium product that's reimbursable, all while offering big opportunities for cost savings in the chronic wound care space, where the average patient cost is already exceeding $11,000. Through our program development, LIMAX will have many exit opportunities that result in high return on investment after 5 years, with many highly viable M&A prospects, as our technology is a clear fit into their product portfolios, including both companies in this competing space, as well as manufacturers and distributors. Our team has close ties to the problem that we're solving. My name is Ben Friedman. I've been working to augment tissue healing for the past decade. My co-founder, Emily, has translated her background in chemistry and material science to enable new solutions to treat human diseases at the Wyss Institute. Our other co-founder, Dave Mooney, has spent his entire career pioneering novel therapies to improve tissue healing and has provided key advising and laboratory funding to accelerate our progress. We're supported by a beastly lineup of Wyss engineers and advisors that are helping us to take this technology to the next level. We've been fortunate to have the support of many clinical champions throughout Boston who provide us important clinical feedback and have conducted preclinical studies. We've also made significant traction in validating our technology at the Wyss with further studies in biodegradation and removal across several indications. And don't just take our word for it. Excitement surrounding this technology has been picked up by over 100 news outlets around the world for our slug-inspired materials. And most recently, we were on exhibit at the Cooper Hewitt Smithsonian Museum in New York City. We have also made traction on the commercial opportunity front as finalists in Harvard's 2019 President's Innovation Challenge and winners of the 2019 Swiss Next Pitch competition. We've also worked with teams at the Harvard Business School and Boston University to develop our business model and have participated in the National Science Foundation I-Corps program, where we've conducted over 120 customer interviews to further validate product market fit. Finally, we've mapped out our path to regulatory approval and have identified mechanisms by which our technology can be reimbursed. Although we've talked today about the treatment of chronic pressure ulcers in growth phase 1, LIMAX is truly a platform technology. We plan to expand applications of our technology to other types of wounds, like burns and traumas in growth phase 2. Together, that's a $1 billion market opportunity. This will enable early revenue streams as we simultaneously pursue approval for our Class 3 regulated biodegradable internal sealant and drug delivery system in growth phase 3. Together with AppMax, a novel applicator device used to carefully position the TissueMax adhesive within the body, and RemMax, our patented system that allows for on-demand triggerable adhesive removal, we have a pipeline of products in development. No one should suffer from a chronic wound. The LIMAX technology addresses unmet needs in ulcer treatment and also has large addressable market opportunity. Our strong IP portfolio enables impact both in topical applications as well as in later growth phases as an internal sealant. And we're currently seeking funding to take our venture to the next level. Together, let's transform care for millions of patients around the world and give Nick the solution he's been waiting for. We'd like to give a huge thank you to Rachel and Yael for organizing our working sessions, and especially to our all-star mentoring team for their incredible insight, support, and guidance for the past 8 weeks. We've learned so much in this short program and are truly grateful for their dedication to the project and time. Thanks again for your attention. I'd be happy to take any questions.
Great job, Ben. All right, you have a couple questions lined up in the chat. And some very good comments. First question. Thank you, Ben. Can this material be used on diabetic wound patients? Has this been tested on such patients?
That's a great question. We'd like to ultimately use it on diabetic wound patients as well. We have not tested it yet, though, in that type of model system.
Great. Question from mentor Joel Kahn. Nice presentation. What type of immune reaction or other side effects are there?
Yes, we've done a variety of in vivo studies so far, as well as in vitro studies to examine biocompatibility of the materials. Overall, we've seen very mild responses after implantation of these materials out to beyond 4 months in vivo. in small animal studies. We're still planning to need to go through the full ISO 10993 biocompatibility test. But so far, everything that we've seen and published has shown very mild and minor responses.
Great. Ben, are you looking at both internal and external applications? For internal applications, is it possible you may be required to follow a PMA regulatory pathway for those indications?
Yes, we are indeed looking at both applications in the multiple growth phases that I was just highlighting briefly at the end of the talk. Early for the topical applications, yes, we're focused on the 510K pathway, but for the internal, yes, it'll be a PMA.
And final question from Abby Barrow. Have you looked at the potential for use in beauty products? There's many snail slime creams and treatments out there.
That's a great question. Yes, after the 2019 President's Innovation Challenge, we were approached by a large company in this space. So we definitely have some interest in that space as well, and be happy to talk more.
Okay, we're getting a lot of questions for you. I'll just choose one more. Does it adhere to non-biological material? How quickly does it adhere?
Yeah, so to tissue, adhesion generated is very rapid. Within the order of a few minutes, we can generate adhesion beyond that of commercially available products, such as fibrin-based glues and co-seal and other internal sealants like that. The material by itself does not adhere to non-biologic properties or non-biologic tissues that would require additional surface modification to make it adhere to those types of materials.
Excellent. Well, thank you, Ben. Wonderful presentation. I will also ask you, there's a couple more questions in the Q&A function if you'd like to answer there. And then again, if folks are interested in connecting with any of these entrepreneurs, you know, they have been listing their email address, but I'm also happy to make any connections as well.
Thank you.
Thank you, Ben. Our next presenters, plural, are graduating from our MassCONNECT DH program today. And the first are Jonathan Wang and Mark Kalinich from Watershed Informatics. Over to you both.
My apologies. Zoom decided to stop the share function on my side. One moment. All right, there we go. So thank you everybody for the opportunity to present here at MassCONNECT. Jonathan and I are extremely grateful for the opportunity. Just to tell you our story. So I'm Mark. I grew up out in Northwest Indiana and initially came to Boston in 2009, where I first met Jonathan. He was in room 413 of Baker House at MIT, and I was in room 411. Jonathan's an electrical engineer and computer scientist, and while he was learning about high-performance computational architectures, I was working in targeted drug delivery systems in Bob Langer's group. After graduating, I went on to go to the Harvard MIT MD PhD program to learn how to work at the interface of the bench and the bedside. And Jonathan co-founded his own quantitative trading firm, Domeyard, that he'll tell you about in a little bit. And it was really during my PhD that we had the core insight for Watershed, that I had generated these omics data sets, some DNA sequencing, some RNA sequencing, some proteomics, some metabolomics, and was ripping my hair out trying to figure out how to do basic data manipulations. And I would look to my friends and academics, and they would tell me this was the state of the field. I would talk to friends in biotech, and they said that they had this problem too, that you couldn't just throw money at it to actually accelerate your science. And even folks in pharma would tell me that they'd have 300 questions and be able to answer 3 of them, as long as the bioinformatician's bonus was based on whether or not that question got answered. And I was chatting with Jonathan about this one day, and after telling him what I just told you, he told me, oh my God, I actually solved a very similar problem for my trading firm back in 2015, and I'll let him take it over from here. Oh, muted, Jonathan.
Hi everyone, I'm Jonathan, and as Mark mentioned, my background is in electrical engineering and computer science, and prior to this, I co-founded a quantitative high-frequency trading firm where I was really in charge of the entire tech stack, and that included everything from building out the low-latency trading engine all the way to managing the machine learning research infrastructure used by our internal research team. And specifically on the research side of things, we had a team of quant researchers whose job it was to look at petabytes of financial data and to use all of that information to figure out how to optimize our automated trading strategies. And just for reference, these people, their backgrounds were in math and physics, and they knew exactly what they were looking for in these data sets. They knew the type of signals they wanted to test out and the hypotheses they needed to go back and forth on. But the problem was, every time they wanted to run one of their data experiments, they would have to sit down with a software engineer who would then go and code up hundreds of lines of code on their behalf just to be able to run a one-off experiment. And this was a problem because it wasn't very efficient, since the researchers were sitting around most of the time waiting on someone else to turn around their data for them. And it wasn't very scalable either, because as our data sets grew, every time we wanted to bring on a new researcher, we had to hire an additional software developer who could sit down with that researcher essentially full time. And our solution was to really build out a set of tools and an entire platform that empowered the researchers themselves to be able to design and run and iteratively execute their own data experiments and to take advantage of all the high performance computing power that we had in-house. And we rolled it out and we saw how successful it was, not only with improving the work efficiency of the researchers, but also in terms of how that translated into better trading strategies for our company. So when Mark and I talked, we realized that for biologists, even though it seems like they're operating in a completely different technical domain, in many ways, the type of problem they're facing is very similar, where even though biologists are asking the questions of the data and are the ones who are most connected to the data, they're constantly relying on someone else to go and actually write out code and to actually do the experimentation for them on the data analysis side.
So let's zoom out a little bit. That over the past 10 years, the rate limiting step to insight has rapidly transitioned from data generation to data analysis and interpretation within biotech and pharma. That biologists who are working to build better drugs faster and deploy them to the right people currently wait months to work with highly oversubscribed bioinformaticians for performing even rudimentary data analysis tasks. Conversely, these bioinformaticians are extremely frustrated by the demands on their time for executing basic analyses rather than the sophisticated bespoke solutions that they were designed or that they were hired to design. At the core of this problem is a mess of tools and infrastructure that currently requires an informatics expert for use. The Watershed Data Lab overcomes this analysis bottleneck by empowering the biologist to analyze and interpret their own data, as well as augmenting the abilities of an organization's bioinformaticians. Watershed achieves this because we've provided the right balance between flexibility and usability, taking the time to insight for these biologists and bioinformaticians from months to minutes. Now, we've built out the core computational infrastructure necessary to achieve this task, as well as one flagship omics offering, bulk RNA-seq, for our minimum viable product. Now, we knew from my own experiences and our market research what biologists needed and provided the feature list that you see on the left. We set up this MVP to test one key hypothesis, that biologists can use and will love using a low-code workflow for analyzing and interpreting their data. And when we went over our game film and watched and reviewed recordings of our users, we made a few key discoveries. The first was that biologists could not only use, but were delighted by the platform, some even describing it as life-changing. That because it was so easy to use, the biologists could discover that they could answer their data science questions in minutes and rather than months. Now, Jonathan and I had hoped the first two to be true. That's why we founded the company. But the third fact that we found out wasn't something that we'd expected, that because it was so easy to ask these questions, that biologists started asking more questions, that Watershed wasn't just a productivity tool for them. It actually widened the aperture of discovery by enabling them to answer questions they never otherwise would have asked. Now, biologists have loved our platform. It's currently rolled out to 7 different groups, both in biotech and biotech and academics. And notably, it's not just useful across academic disciplines, whether or not you're in psychiatric genomics or cancer epigenetics, but also across the computational skill set spectrum. It doesn't matter if you've been coding for longer than I've been alive, like Roy Perlis, or have never opened the command line in your life, like Vinod. Everyone has found benefits to using the Watershed platform. Now, if we take our science hats off and put our business hats on, we saw that the overall growth in the data storage and compute for single and multi-omics data sets across the academic, biotech, and pharma ecosystems, we see that the moderate $3.5 billion 2020 market is going to balloon to a $40 billion market opportunity by 2025. Now, of course, we weren't the first, second, or third teams to recognize this major opportunity. But other folks have tried tackling it in a few different ways. Fundamentally, watershed is contrarian in our thinking in two major ways. We think that biologists are the true customer, and that the vast majority of biologists actively analyzing these data either have learned or are equipped to quickly learn basic high-level programming environments like Python, R, or MATLAB. Now, to dive a little deeper into the competition, you can see that there are two major axes here: flexibility on the x-axis and usability on the right. Products like FireCloud and Nextflow provide the flexibility needed to answer a biologist question. This is in the lower right, but at the cost of requiring an undergraduate computer science degree. Conversely, Illumina's Sequence Hub, DNA Nexus, and others have built easy-to-use products. This is in the upper left, but at the cost of not being able to provide the customizability required to truly answer a biologist's questions. Watershed hits the sweet spot of on the flexibility usability curve. Uniquely, Watershed is set up to be a fully integrated vertical solution that addresses all parts of data storage, compute, and analysis for our users. Now that we've built out our Watershed core, we're adding in adjacent workflows over the next 18 months to provide the full stack solution that our customers across the academic, biotech, and pharma ecosystems need. Now, we've assembled the stellar team in order to do that. We've told you about Jonathan and myself, and we also have Alvin, who was the lead developer of Google Hangouts Chat, as well as a co-developer of Materialize, and Jonathan Terry, who has expertise in bridging the software-hardware gap. We also have a set of world-class advisors, including Frank Moss for software strategy, Daniel Haber for biology insight, and Michael Lawrence for informatics expertise. Now, our goals for this next round of funding are to achieve a full stack, scalable, integrative multi-omics support, have product market fit in both biotech and pharma, and achieve significant academic key opinion leader usage. We're hoping to do that by getting 2 pharma clients, 7 biotechnology startups, and 20 academic researchers across 5 different groups. We have the product milestones that we discussed here, and actually, as of today, are happy to announce that Bessemer Venture Partners will be leading our next round, our next $3.5 million round of financing, although we are looking for follow-on investment. Our long-term vision is to create the additional single omics workflows that we've needed to support our multi-omics data analyses, and then dominate and become the de facto standard for biologic data analysis. And with that, I'd like to thank everyone from the bottom of my and Jonathan's hearts for helping us out for the past 6 weeks. You guys have been transformative in the way we approach things, from go-to-market to thinking about our customers to our financing, and we couldn't be happier to be part of the MassCONNECT family. So with that, we're happy to take any and all questions that y'all might have.
Great. Thanks, Mark. Thanks, Jonathan. Again, opening it up for questions to these two. You can use the Q&A function at the bottom of your screen, like I know you have been doing. We'll leave it open. Ben, you still have one unanswered question in there for you. First is a comment actually from your mentor, Paul Hartung. Congratulations on the financing.
Oh, thank you, Paul. We were very pleased to get that email earlier today.
Couple more seconds.
While we're waiting for those questions, I did want to also put in a plug to any potential users. We're very actively seeking out folks that are interested in working with us as we co-develop our next workflows. So if you happen to have bioinformatics needs, please don't hesitate to reach out.
I think that your presentation was just very crisp and clear.
Good to hear. Well,
let's see. Another comment, very impressive presentation from someone that was on your mentor team, Kirk Facker. All good feedback. All right, well, I will ask you two just to hang out in case there's any other questions. Here we go. One last one is from Super Mentor, Vineet Najalan. Do you need syndicate investors?
So the round is currently oversubscribed. However, we are very interested in hearing from investors that might be strategic as we move forward. So please do reach out.
And one other question. How did your MVP compare with traditional RNA-seq analysis?
Great question. So Watershed doesn't hope to build any new algorithms or optimize a specific implementation. What we try to do is radically democratize access to these existing workflows. So for our users, we want to make it so that you can easily upload your data, do the analysis that you know you want, but don't speak the low-level computing languages in order to execute on, and then get your plots in minutes rather than months. So for instance, our users want to do PCA, overlay their metadata on whatever their exploratory plots are, do some differential expression, do gene set enrichment analysis, the standard issue workflows. But now the biologist feels empowered to actually perform these without having to wait months for the bioinformatician on back and forth. Happy also to talk about how it also serves as a medium of communication between the biologist and the bioinformatician.
Excellent. All right. Well, again, any additional questions for these two, they will be sticking around to answer in the chat. Thank you, Mark, and thank you, Jonathan. Great job.
Thank you.
And last but not least, we finally have Julieta Gleiser from Step Up IC and Alchemy CGI, the other graduate from MassCONNECT DH.
Thank you, Rachel. Thank you, Rachel. I'm going to start by sharing my screen. Here we go. Thanks again. Thank you so much for the MassCONNECT team for making this possible and for giving me the opportunity to share with you our project today. I'm here to talk about Step Up, our digital therapeutic solution for patients with intermittent claudication. Step Up is a remote monitoring and automated coaching digital therapeutic app for patients with IC. The platform harnesses behavioral sciences, machine learning, and predictive modeling to detect risks of complications and provide real-time personalized interventions. Intermittent claudication is the most common presentation of peripheral artery disease, a chronic ailment that affects 200 million people worldwide and approximately 12 million people in the United States. It's caused by buildup of fatty plaque in the arteries in the lower extremities. It's extremely painful, and if left untreated, it can cause limb loss. In layman terms, it's like a heart attack in your leg. The artery gets clogged. There is no circulation or poor circulation. It creates ischemia, and it needs to be treated, or you can have complications that go beyond into cardiac problems or stroke. Currently, there are two main forms of treatment for IC. The most commonly used is revascularization by placing a stent or a bypass. This is a very common way of treating the disease, but it's actually not recommended by the Society of Vascular Surgeons because it yields very short-term benefits. If the patient doesn't change certain habits, they will have to be revascularized in the next 2 years and so on until their limb becomes compromised. The other option of treatment, which is actually the first line of treatment for this type of disease, is walking therapy. The issue with walking therapy is that if the physician sends the patient to walk without any behavioral insights or without any behavioral guidance on how to adhere to this type of treatment, it's highly unsuccessful. There is supervised exercise therapy available for patients, but it's very low prescribed in the United States. Only 2% of physicians prescribe this type of therapy. And even when they do, there are other issues such as inaccessibility. The patient has to actually go to a clinic, or maybe the clinic does not offer supervised exercise therapy specific for patients with peripheral artery disease. As a result, a treatment that is fairly low cost at $7,000 a year compared to another treatment that is close to $14,000 a year is not highly used in the United States. We're trying to come up with a solution that can revert this and prevent unnecessary vascular interventions. Our app brings high value care via remote monitoring and automated interventions. And the way it works is it gathers objective data from the patient, whether it's walking trends, how many minutes they've walked, time of the and pain onset, and quality of life, and other types of activity collected by the app. This is combined with other subjective data, and it goes through a predictive model that can detect when there is the risk of complications. In addition to this, there is an automatic and personalized system of interventions where patients receive time and personalized nudges informed by behavioral sciences. The third aspect of our app is that it can alert the physicians when a patient is at risk of developing complications or when the app notices that the patient's disease is advancing. Part of the way we detect this is through this remote monitoring, but it has an added benefit that the patient can do supervised exercise therapy from the comfort of their home or wherever they choose. They don't have to go specifically to in-clinic therapy. I want to talk a little bit about the market. Currently, our target market is the Medicare intermittent claudican population that accounts for around 2.4 million patients in the United States. Our total addressable market in the year 2020 is approximately $21 billion. This is primarily caused by the high amount of surgeries performed. The mHealth market is growing exponentially and is expected to reach $102 billion by 2023. And within this large market, the digital therapeutic market, which is the one that we fall into, is expected to reach $6.9 billion by 2025. Our advantage in this competitive market is that the category of remote patient monitoring with the digital therapeutics is the one with least competition. So how are we going to implement this? We've been considering different paths. The first path that we are looking into is to partnering with a pharmaceutical company. In this context, our app would serve as an augmentation to adherence for a prescribed drug. It would be prescribed in combination with a molecule, whether this molecule is already existing or is a new molecule. For example, a statin, which is a medication that the patients typically receive as part of their treatment. We could pair our digital therapeutic to a statin in order to enhance adherence and create other benefits to the overall patient therapy. If the app is paired with a new molecule, it could also lead to new IP for the combined therapy. We've been exploring FDA paths for this option, and the 505(b)(2) option opens up a direct path to FDA approval. The potential revenue that we have come up with based on our revenue models is around $1 million for the first year after deployment. Another option that we've been considering is partnering with ACOs and Medicare Advantage. ACOs are accountable care organizations. It's a group of physicians that team up to provide value-based care. So this aligns 100% in line with our digital therapeutic. We would offer a flat fee per month per patient. It would be prescription-based, and there are a bunch of codes that would cover reimbursement for physicians if they want to prescribe the app and use the remote monitoring system. The potential revenue for this model is projected at $3 million by 2025, given by 2025. A third option that we are looking into is partnering with medical devices. The context of this partnership is similar to the pharmaceutical, where the app would enhance and enable benefits for an existing medical device or a new medical device, such as Medtronic's Guardian. It's a glucometer that is combined with an app currently, but could have additional features specific for the diabetic population. And the diabetic population is important because most patients with PAD suffer from comorbidities such as diabetes, so they could be tackled together. In terms of timeline and financing, we are hoping to get $500,000 for the first 6 months. This would allow us to finish up building our minimum viable product and deploy a test on 240 patients. We would be doing this in collaboration with Mass General. We have a collaboration with them actually that I'm going to talk about in just a minute when I go to describe my team. This first 6 months also would allow us to build a predictive algorithm that would gather data and have basic capabilities of predicting disease progression. And then in the second phase, in the stage gate, we would need $1 million. This is to develop a machine learning algorithm that would add digital phenotyping and comorbidities and much more data individualized for each patient that could make the predictive model much more specific and customized to each patient. Once we have this app, we would like to, as our long-term goal, to be able to replicate and expand this predictive model using digital phenotyping to other chronic diseases, where early intervention, risk prediction, and behavioral insights can reduce complications and avoid unnecessary interventions. And we are currently exploring a similar approach to treat diabetic patients with neuropathic foot pain as another alternative to our product. I'd like to talk a little bit about who we are, who is the group that is trying to take this effort forward. Very quickly, I worked in the film industry as a visual effects producer for 15 years in movies like Iron Man and Pirates of the Caribbean. Later on, I moved to develop digital content for TV for children and adults as well, and also the development of applications. And 3 years ago, I moved to Boston and decided that I wanted to put my skill sets to work in a different area. So I started a company developing applications to help patients. I teamed up with Dana-Farber to develop a pain management application for patients with cancer that is currently under development. And this year, I also joined at the beginning of the year Mass General's incubator, MESH. Through MESH, I met Dr. Anaita Dua, who is instrumental in the development of STEP-UP-IC. Dr. Dua is the director of the vascular lab. She's also an assistant professor of surgery at Harvard, and she's a strong advocate of reducing unnecessary surgeries in patients with PAD. So she is really pushing to get this project to patients as soon as possible. Jacqueline Lefkowitz works in behavioral sciences and has experience in developing interventional apps and behavioral design to change patients' behavior and to help with patients' behavioral insights. And she's an MPP candidate at Harvard. And Dokechi Bombs is an MPP MD candidate at Harvard, who is not only helping with the medical side of the app, but also with development of the predictive algorithm. A huge thank you to MassCONNECT, Rachel, and the rest of the team at MassCONNECT. The mentors have been amazing. I can't thank you so much. This has been a wonderful experience. Thank you for your insights. Thank you for your time. It's been a pleasure to work with you. Thanks so much.
Excellent. Great job, Julie. We will now open it up for questions, again, from the audience. Again, in the Q&A function. Any questions for Julie? We'll wait a little bit. I'm also getting questions on my phone. Julie, can you speak to the IP situation for a step up?
Sure. So in general, software for applications is not patentable. There is no IP for software as a intellectual property itself. What we are trying to capitalize is on the machine learning predictive algorithm that we intend to use after the first phase of deployment of the app. This would still be a trade secret, so it wouldn't be patentable.
And then can you speak to the regulatory pathway?
Sure. So let me go back a little bit. If we were to team up with a pharmaceutical company, the way that the model we expect would work is we would have a FDA approval for a therapy, regardless of the FDA approval for the drug, but they would be joined together to create one product. Now, the specific pathway for the digital therapeutic app, what I mentioned, this 505(b)(2, what it means is that the app would be based on evidence that already exists. A lot of the trials have been done in the lab. It is known that patients benefit from supervised exercise therapy over surgery. There is data that backs that. There's data that backs that patients do much better when they are supervised and coached than when they try to do the supervised exercise therapy alone. What hasn't currently been built is the app itself to do it remotely from anywhere without having it in a lab setting. So the path for FDA approval would be much faster.
Excellent. Well, no other questions in the box yet. I know we're 3 minutes over, which is still good by all accounts. So I think we'll leave it there, Julie. And you had your contact information up as well.
Yes, let me put it back. There we go.
Should anyone want to reach out to her directly. Thank you, Julie.
Thank you, Rachel.
All right. All right, and congratulations. Congratulations to all of our presenters and an official welcome to the MassCONNECT family. It has been so much fun to work with each of you and to witness your growth in such a short period of time. We hope that you'll stay in touch with your mentors and with us here at MassBio too, because you are now also part of the MassBio family, as we are pleased to offer you all a free year of MassBio membership. Our program for the afternoon now concludes, and you will be able to network with all of our entrepreneurs and sponsors tomorrow at 4:00 p.m. For the MassBio holiday, the virtual holiday mixer. Again, if you haven't registered, I think the registration is closed, so please ping me and I'm happy to sign you up on the back end. As for MassCONNECT, we are taking a tiny hiatus until February, and we'll kick off with another round for our traditional cycle. And then in April, we'll start the digital health cycle. I hope to see you all tomorrow at the networking in your holiday gear. But if I don't, I wish you all very happy holidays and a very happy new year. Take care, everyone. Good night.